Welcome to our presentation for Group 1, MIC 211 Microbiology Group, Gram stain techniques. Alright, the name Gram is gotten from a Danish bacteriologist called Hans Christian Gram in 1883. Alright, so the Gram staining techniques are divided into two, the Gram negative bacteria and the Gram positive bacteria, and they are differentiated according to their cell wall constituent. Okay, so the gram-positive cell wall is made up of peptidoglycan layer, which is a thick one, and the negative one has a thin cell wall. Okay, so moving to the next, let's look at the principle of gram staining techniques. The gram-negative bacteria and the gram-positive bacteria. We're going to be making use of some chemicals. First and foremost, we stain both of them, both negative and positive bacteria, with crystal violet, and then it gives a violet color. And also next we stain with gram iodine, and it retains their color, which is violet. Don't forget, gram iodine is called mordant. And also next we stain with methanol, and when we stain with methanol, which is called the decolorizer, only the gram negative bacteria will decolorize, while the gram positive bacteria retain its color violet. Okay, so next we stain with counter stain, which is called carbol fulchine, which is called counter stain. Okay, so we find out that gram negative bacteria change to pink color, while the positive retains color or become deep violet. Okay, all right, the reason is because of their differences in their cell wall. Gram-positive bacteria has a thick peptidoglycan layer, and the gram-negative bacteria has a thin one, and the outer membrane is thin and also pink. Okay, so at the end product, the primary stain for the positive bacteria is violet, and the end product of the negative one is pink. So you have to know that. Go to the next one. We have the preparation of bacteria smear. Okay, all right, the requirements, we have the bacteria culture plate, the inoculation loop, the distilled water, and also have the spirit lamp. We have the microscopic glass slide. Those are the working with for this practical. So the steps: first, ignite the spirit flame, and then you flame the inoculation loop, and then you allow it to cool. Okay, then next, you take a clean microscopic glass slide and place a drop of sterile distilled water on the clean glass slide using the inoculation loop as a help. Okay, then next, you take a small portion of the bacterial colony from the agar plate, and then you emulsify it with a drop of water on the glass slide. Okay, and then you spread the smear to the size of about 1 centimeter square. All right, as you can see the video, and then you allow to air dry. After that, you allow it to air dry. Okay, allow to air dry for about 2 to 3 minutes. All right, and then after that, you pass it over the flame gently. Pass it over the flame gently. Okay, that's what they call its fixation of smear. All right, next is the procedure for gram staining. And let's talk about the material required for the process. Now, the first one, we need crystal violet solution, and then followed by the grams iodine, and then we need the methanol, then we need the dilute carbon fusion. Okay, those are the things we need. Apart from that, the other things we need are the distilled water in that white container. We need the distilled water, the heated fixed smear, the blotting paper, and the staining rack. All right, these are what we're going to be working with. Okay, next step. So we place the heat fixed slide on the staining rack, and then we add crystal violet, which is the primary stain. Okay, as you can see it, and then we wash off with the distilled water. Okay, after you have left it for about 2 minutes, you wash with distilled water gently. All right, so the next step, we add grams iodine solution to the smear. Okay, we leave for about 1 to 2 minutes, and then after that we wash off with the same distilled water. Okay, after that we add again methanol over the smear for about 15 to 20 seconds. Okay, methanol is very powerful, so you reduce the time 15 to 20 seconds. Okay, and wash off with the still water. And after that we add dilute carbol fusion. You leave that also for about 30 to 60 seconds, and also to you wash off with the distilled water. Wash off with distilled water, and then after that you blot dry with the blotting paper. You blot dry with the blotting paper, and then you place the stain slide under the stage for microscopic examination. Okay, all right, you use the microscope. You follow the order. Okay, okay, examine under 100 times. Okay, under the electron microscope. All right, so the gram staining, when you find out you have this kind of color violet. Okay, if it's violet color, that means it's gram positive, according to how we explained the other time. If it's gram color, violet color is gram positive. And also if it is spherical shape, then it is cocci. Okay, if it's a spherical shape, then bacteria is cocci. And also if the rings are arranged in clusters, then it is staphylococcus species. Okay, so you call it staphylococcus species. Then it is still gram positive. It is still gram positive, but with different area. Okay, you can see it is arranged in rings, and if it is arranged in rings, it is still, don't forget, it's still violet color, grams positive. Okay, so you call it streptococci. You call it streptococci species. Okay, that is for grams positive. Alright, so you can see we have the two type of it. But also, if you have it like this in this pink color, all right, you call it gram negative bacteria. That's a gram negative bacteria. Okay, and also if it has rod shape, like the way you are seeing it, if it's a rod shape, then it is bacilli. And also if it has no specific arrangement, then you can call it E. coli or Salmonella, etc. Okay, so those are the gram-negative bacteria we have. All right, all right. Thanks for watching. Bye bye.